Again, hi, I'm Sandra Mosquera. I'm part of the Department of Plant Production and Forest Resources, and I'm going to give you today an overview of the delivery of double-stranded RNA and silencing RNA in RNA-based technologies for the control of plant pathogenic fungi. So first, let me explain you a little bit what is the RNA interference. From now on, RNAi. This is a cellular mechanism that is present in most eukaryotic cells. And basically what this mechanism does is that it uses double-stranded RNA to control gene expression through translation and transcription and repression. So how does it work? Basically, what you need is a double-stranded RNA that can be produced either by the nucleus of the cell or can be like given to the cell externally. And then this dicer protein, which is also produced by the cells, is going to cut this double-stranded RNA to make small RNAs or microRNAs. Then these small molecules are going to join these other proteins in the cells, and they are going to form what is called the RISC complex. And then this RISC complex is going to use one of these strands of RNA and are going to target messenger's RNA to downregulate the expression. It can do it, it does it in a sequence-specific manner, and it can just like cleavage your messenger RNA, so then not protein is going to be produced, or it can be done translational repression. There is some, like, why is this really important? This can not only happen in one cell. Actually, these RNA signals serve as a mean of communication between cells. So the silencing process that is happening in one cell can go to the entire organism because it passes from one cell to another. And from what is that important to us? Because actually plants use this RNA to defend themselves from pathogens, since the signals can not only translocate between cells of the same organism, but it can translocate from plants to the interacting pathogen. And that is something that is known as cross-kingdom communication between the plant and the pathogen. So how can we make use of these cellular mechanisms? So basically, we can like modify plants to express double-stranded RNA specific to pathogens and control it by downregulated like pathogenic genes. And how does it work? So as I told you, like the double-stranded RNA can be produced by the nucleus of the cell. So here is what happened. And then these molecules go into the fungi where they are processed by the fungi machinery, and they will cleave the specific gene that you want to silence. This is something that is known as host-induced gene silencing. These have been proven before, like here I just show you an example. So for example, this is an example of his working in Fusarium graminearum in Arabidopsis. This Arabidopsis had been transformed to express a double-stranded RNA of this pathogenicity gene in Fusarium. So here what can we see is that while the wild type, the infection of Fusarium is like going normally, when you have Arabidopsis plant expressing this molecule, this double-stranded RNA, you see a reduction of the infection. Here you can see like a reduction of the area, and you also see how these genes are, the expression of these genes are actually downregulated compared to the control. Okay, so what are the main challenges of this Higgs technology? Is the first, some plants are not easy to transform, so you have difficulty of getting actually the plant to produce this molecule. And also, these plants, they have a low public acceptance because of their GMO. So, as an alternative, as I told you before, you can also, like, not necessarily a plant need to produce it, but you can, like, give the plant externally these molecules. So, we basically can just apply the molecule, the double-stranded RNA, that is specific to a pathogen to control and that regulate these genes. This is something that is called spray-induced gene silencing, or SIGs. Here we see in this paper an example of the SIGs working in Fusarium graminearum on barley leaf. These leaves were sprayed with this double-stranded RNA. Again, this is specific to pathogenicity genes in Fusarium. Here you can see like the infection in the treated plants are almost like you don't see much symptoms, while you see symptoms development in the control with the buffer and with a GFP double-stranded RNA that is not specific to the fungi, so it shouldn't affect the pathogen. So also we see like what we are seeing here in the picture, just the reduction of the symptoms and also the downregulation of the genes. So basically what they show in this paper, and you can reduce the infection and gene expression in barley leaves just by inoculating this double-stranded RNA that is specific to pathogenicity genes of this fungi. So this technology is promising, but it also has some challenges. What are some of the challenges of the SIGs? Basically, the double-stranded RNA, the RNAs are not a really stable molecule, so they have to deal with harsh environmental conditions such as nuclease, UV, pH, rain, and that can degrade your molecule, and if you don't have the molecule, you don't have the control. Here, for example, we see a field evaluation in soybeans in this paper. What they did is they applied this molecule, this double-stranded RNA, and you can see how the concentration drops as the days post-inoculation go on. So even though this technology is really promising, and there was like many fields, researchers in many fields have like worked to develop technologies to overcome these obstacles. For example, this has been really developed in the medical field for RNA therapeutics, and one of these technologies is the use of nanocarriers. There are several nanocarriers here, like some examples, the lipid nanocarriers, carbon dots, like there are like different matrices that they are using, or formulation that they are using for like overcome these problems. And what they have found that nanocarriers can actually improve the double-stranded RNA stability, uptake, and therefore increase the silencing of the genes that you want to silence. These have not only been working in the medical field, like we are working slow from the pathology field, but some progress have been made. For example, here in this paper, they are using like lipid nanocarriers, and they are trying to silence genes of these B. germanica, which is like a cockroach. This insect in his midgut juice and hemolymph, they have a lot of nuclei. So what they wanted to try or to evaluate or to prove is whether these lipid nanocarriers will protect the double-stranded RNA from degradation, and they actually find that. So here, for example, you see that when the double-stranded RNA is naked, you see a complete degradation. But then when you have conjugated with the liposome, you don't see the degradation. And they also prove in time that the stability of the molecule is improved. On the other hand, in this other paper, they use a different carrier and a different, like this is a pest, but this is a pathogen, this is using Phytophthora. And they use carbon dots, and they actually find that the carbon dots increase the silencing capacity of the double-stranded RNA. For example, here you can see the GFP control. Here you can see that when the naked RNA is applied, you don't see a control, but then when you have the double-stranded RNA protected by the carbon dots, then you see like an increase in efficacy of silencing. So in this last paper, they just show that there is an increase, like a better protection, but they don't know if it's whether because the uptake is improved or whether the double-stranded RNA is protected. And actually, that is one of the other problems of this technology. These molecules need to pass through a lot of barriers to like get to the fungi. So, for example, they need to go through the plant cuticle, they need to go to the plant cell wall and membrane, and also the fungal cell wall. So we don't know how this is happening, but we have like some knowledge about this, and I'm going to just quickly tell you like what we know. Basically, what we know is that when you have a pathogen that is susceptible to silencing machinery, the efficiency of the silencing is going to depend by the fungal uptake. We also know that the claritin-mediated endocytosis and the extracellular vesicles are involved in the uptake by the fungi. For example, in this paper, when you use like chemical inhibitors of the endocytosis, you see a reduction of the silencing. And also in here, you can see with the GFP labeled vesicles that the double-stranded RNA is actually going inside the fungi. So, considering all of this, what we know so far is that nanocarriers protect double-stranded RNA and facilitate its delivery, and also that vesicles facilitate the double-stranded RNA uptake by the fungi. So knowing that, it looks like a promising idea or like a good idea to use extracellular vesicles, in particular bacterial vesicles, to like use double-stranded RNA mediated control against fungi and oomycetes. And actually that is what we want to do in, like I'm going to begin, I'm working in the pathology forestry lab. This is the lab of Julio Diaz, and we are working in a project to use this technology. The project is named Supera. And this is the objective of this project. We are going to use this extracellular vesicle with double-stranded RNA molecules to target fungi and oomycetes, in particular this one, and in this host. Here I just put like the name of my team because I'm not working alone in this one, and basically I'm out of time, sorry, so like it was 10 quick.